Hello, my name is Vadim Karnaukhov, and my work is dedicated to the development of a statistical pairwise potential for prediction of T-cell receptor peptide recognition. T-cells are the key players of the adaptive immune system. They can recognize foreign antigens and kill cells carrying these antigens. Specificity of a T-cell is determined by T-cell receptor. I will further call it TCR, membrane-anchored protein, which recognize peptides presented by special proteins called magic histocompatibility complex or MHC. Each of us has hundreds of millions of T-cell clones with different specificities. Currently, using high-throughput sequencing, we can obtain sequences of TCR repertoire of each individual and even find clones associated with some conditions: infections, autoimmune diseases, or cancer. However, it's difficult to find out which antigens, which peptides these TCRs recognize. Prediction of TCR specificity is critical for a number of applications in cancer immunotherapy, vaccine design, and autoimmune disease treatment. In this work, we aim to develop a statistical potential for prediction of TCR peptide recognition, and we name this potential TCRen. Why can't we use for this task traditional approaches for exploration of protein-protein interactions? The reason is that TCR peptide interface has a very special nature. Other protein-protein interactions, for example, enzyme inhibitor complexes or protein dimers, are in general characterized by a high affinity and are mainly mediated by non-specific hydrophobic interactions. On the opposite, biological functions of TCRs require low affinity and more specific interactions mediated mainly by polar amino acids. That results in depletion of hydrophobic and enrichment of polar amino acids in TCR contact regions. At the same time, peptides may be regarded as a sample from proteome and have amino acid composition close to proteome 1, and this leads to asymmetry of TCR peptide interface in terms of amino acid composition. So, in derivation of TCRen, we focused only on amino acid contact preferences in available TCR peptide MHC crystal structures and designed our potential as asymmetric, with an equality of residues of the same amino acid in TCR and peptide site. For derivation of TCRen, we collected a non-redundant set of TCR peptide MHC complexes from PDB, extracted all pairwise contacts between amino acids in TCRs and peptides, and computed amino acid contact preferences. TCRen may be represented as a matrix with 20 rows and 20 columns, and each cell corresponds to a value of pairwise interaction energy between the given two amino acids. There is a number of other amino acid pairwise potentials which were derived from statistics of amino acid contact preferences in general protein-protein interfaces, not restricted to any special case such as TCR peptide interaction. All of these potentials are very similar, and in all our benchmarks had similar performance. So for simplicity, I will demonstrate here only one representative potential from the work of Keshkin, which reflects amino acid contact preferences in general protein-protein interactions. We see that Keshkin potential is symmetric and has some intrinsic regularity with clear clustering of polar, nonpolar, polar, and charged amino acids. On the opposite, TCRen has no regular pattern and it's asymmetric. We tested performance of TCRen on several benchmarks. We demonstrated that our potential can accurately distinguish cognate peptide recognized by the given TCR from a set of unrelated peptides. In this benchmark, other pairwise contact potentials, such as Keshkin, work like a random classifier. We also compared performance of TCRen with Rosetta scoring function and MMPCA approach on a test case of DMF5 TCR, which recognized two groups of peptides with different motifs, with hydrophobic and with charged. To do this, we performed large-scale molecular dynamic simulations of length 100 nanoseconds for complexes with different peptides, split the trajectories to 1000 frames, and averaged energy of interaction across these frames. Only TCRen was able to explain recognition of both groups of peptides, while Rosetta and MMPBC approach could not do that. Finally, we demonstrated how TCRen may be used for identification of cancer neoepitopes, which is a very important task in development of cancer immunotherapies. Here you can see a scheme of pipeline that we propose for that task. First, from exome sequencing of tumor and healthy tissues, we identify potential neoepitopes, mutated peptides that are predicted to bind to host MHC. In parallel, TCR sequencing of tumor-specific T cells are obtained. And the question is, what neoepitopes are recognized by these TCRs? Using homology modeling, we obtain structure of TCR peptide MHC complex for these TCRs with a random peptide, and then in silico mutate this random peptide to each of candidate neoepitopes, calculate energy of TCR peptide interaction using TCRen, and rank peptides based on this energy. Top predictions should be further experimentally tested. Using data from previously published work, we performed proof of concept validation of our approach and demonstrated that TCRen can accurately identify the correct neoepitope recognized by tumor-infiltrating lymphocyte. That underlies perspective of TCRen use in the task of identification of cancer neoepitopes. In this slide, you can see conclusions from my work. Thank you for your attention.